Today I'm going to talk about the bold global signal and its role in lifespan research. This is something I've been kind of working for a while now. I've got some new data, some new analyses. Taylor and I worked out this temporal ICA denoising. I want to show you guys. So talk about the global signal first. Essentially, the global signal in bold fMRI research again is we're just getting the average signal across the whole brain. So we're just going to average across all the voxels, and we get one single time course for the entire brain here. And the reason why people are interested in global signal is because the global signal also captures a lot of noise artifacts. So I'll talk about what are the noise components of the global signal. And in order to account for these global artifacts, people have started regressing out, or they started to regress out the global signal. But the problem is that, by definition, the global signal also contains all the neural activity in it. So if you regress out the global signal, you're also regressing out neural activity along all that noise. We've seen out a paper throwing out the baby with the bathwater, essentially arguing against global signal regression. So we'll talk about that, and then a new technique that Glasser and Washu came up with, the Van Essen lab, where they do spatial and temporal ICA denoising to get rid of more of this global noise while keeping global neural signal intact. Talk about some research in the global signal topography, and that's basically where you correlate the global signal with each voxel in the brain. So using the whole brain as an ROI, essentially. There's a lot of new research coming out on that. And then going into the current study where we're going to look at this, how this global signal topography changes across the lifespan, how that's influenced by global signal regression, scrubbing, and this temporal ICA denoising, and then look at how these denoising techniques influence lifespan functional connectivity trajectories. So we're going to go through all of this. First, the global signal, what's in it, a whole bunch of stuff. Physiological motion, scanner drift. So this is trying to turn that out usually. Motion, physiological processes. Motion, so head motion, of course. Cardiac, so vascular system things going into it. Respiration, vigilance. So all of these different things here have been shown to contribute to the global signal. Here. Also coffee and time of day, or I didn't put the citations, but those have also been shown to influence the global signal. So the global signal, again, is filled with a bunch of noise, but we also know, again, that it's filled with also the average activity across the whole brain. So people have started correlating the global signal with different types of processes, or correlating the global signal against itself for denoising techniques. So here's one where they look at the global signal in relationship to EEG vigilance, and they show a high correlation or a strong correlation between vigilance and the global signal. So whenever someone is aroused or becomes vigilant, increases their arousal, that corresponds with the global signal. The global signal is also strongly associated with a couple of these veins and the sagittal sinus signal. So these noise signals in there. And if you regress out some of these processes from the global signals, this is what you end up with. So sort of your unprocessed signal here, minimally pre-processed. And then as you regress out things like motion, physiological things, and then white matter, cerebrospinal fluid, eventually you get left with this little tiny signal down there. So after all that, about 7% of the variance of the global signal remains after you regress out all that. So global signal, again, large contributions from noise, contributions from vigilance. Another thing that influences the global signal is head motion. So these are called carpet plots, and essentially these are all the voxels across the brain. If you look at these up here, they're showing FD, head motion across the scan here. And you can see whenever the subject has a big jump in head motion, there's a big jump in the global signal, and then all the voxels sort of line up. So you get these little lines in here from head motion spikes. The interesting thing is, though, you still get these global signal sort of stripes, even if the subject doesn't have any head motion. So there is a large neural component of the global signal here, shown obviously, because head motion is not causing all these little stripes. But head motion also has a large influence on the global signal, where you can see where head motion happens, there's a lot more of these sort of spikes in there. So see all these little spikes are related to head motion, global signal, value, things like that. Other extra neural component of the global signal, Tochi and Chang, in macaque monkeys, essentially what they do is they inactivate parts of the basal forebrain. And they show that by inactivating parts of the basal forebrain, it influences the time series of certain areas in the brain. So here we see this area TEO, I'm not sure where that was, but when you inactivate one, you see one of the time series jumps. You inactivate the other hemisphere, you see the time series jumps. But for this area 23B, inactivating these left and right areas don't influence it. And then here you see that inactivating the left or right has a large effect on how much variance the global signal explains. So you can see that when you inactivate the left, suddenly the variance explain goes down. You inactivate the right, suddenly the variance explain goes down. So showing a neural component of the global signal there related to the basal forebrain. We have this research group. We started at OHBM where we started looking at global signal stuff, and essentially we showed that the global signal in the HCP data is related to this positive negative axis of behavior. So Steve Smith sort of showed this in a Nature brief communication or whatnot, that functional connectivity strength was related to this positive negative axis. And in this paper, we showed that the global signal is also related to this positive negative axis, which isn't surprising if we consider that the global signal is the sum of all activity. So if functional connectivity is related to something, stands to reason that global signal might be related to it in some way. Taylor just had his paper came out in Nature where he showed that the global signal is actually driving this pattern, this dynamic pattern that accounts for a large part of resting state variance, essentially capitulating the positive negative anti-correlation, I should say. Let's see. So the global signal is involved in a lot of neural processes and a lot of noise processes. And the question is, what do we do about that? So one thing you can do is just regress out the global signal. But again, we don't really want to do that because by definition, you're regressing out brain activity because it's in the global signal. Another thing people have tried to do is scrubbing. So basically deleting frames, which are high in motion. And the thought behind that is that if you delete these high motion frames, and you're going to get rid of all these spikes, which is true. But you're also going to disrupt your temporal continuity of your time series, which you don't want to do sometimes. And then finally, this newest technique came up by Glasser, where they do spatial and temporal ICA denoising. And they essentially claim and show that it's sort of the best way to denoise your data without regressing out the global signal. So getting into these, how do we account for it? I just talked about that, regression, scrubbing, problems with that, disrupt or your regressing neural signal. Global signal regression also distorts your correlations. I'll show you a little thing just a bit. And then scrubbing disrupts your temporal continuity. So the hope is that this new temporal ICA denoising is going to help us get rid of all this noise, but keep time series intact and keep all of our neural signal in there. Global signal, influence of scrubbing. So, oh, not global signal, head motion, I should say. All in one regard, that's just head motion stuff more. So if you look here, this is the influence of global signal regression here. So this is a subject here, and if you do global signal regression here, this is going to get rid of some of those lines.
Jason,
Yes.
could you like put up the laser pointer, or is it that you're not able to?
Oh
see it?
shoot, I've been doing this and you guys haven't seen my arrow this whole time. How do I do this?
Is it not showing? Oh, there's your cursor.
Oh, we can
Oh.
see it now.
Wait, it wasn't up there this whole time? Okay, well, I've been
I
playing
haven't really like looking for it, but yeah, I haven't seen it.
okay, I've been pointing it this whole time at nothing. Okay, anyway.
No, I mean, honestly, it was like two big pictures on the screen, so you could kind of follow what you were pointing to.
Wait, why is it cursing? Okay, but anyway, here's the effective global signal regression. So it's taking out some of these lines here. Doesn't work as well so for some really high motion subjects. And then these people introduced this. I'm not going to get into it, but it's sort of an iterative global signal regression. So the idea is to get rid of all these stripes essentially, and they're showing that global signal doesn't quite do it like their technique does. What else is bad about the global signal? Global signal regression distorts your correlations. So these are your time series without global signal regression. And then you see if you regress out the global signal, it's going to distort your time series. So it's sort of widening this period when the bold signal is in peaking. Whereas these are pretty sharp peaks, it turns them more into these spread out sort of things here. And then you can also see that when your global signal regress, it's going to push essentially most of your correlations, force it to center around zero mathematically. So that is going to also influence your distribution of your correlations here. And you can also see it pushes them together. It not only centers it around zero, it's going to sort of push them all together. So you see here, there's two peaks, and now there's just sort of one peak. And then these people showed global signal distorts correlation patterns also. Forgot the figure for that, but
Wait, sorry, so then the implication with it all being centered around zero is that you might get like false correlations now between brain regions, or I don't understand.
well, not necessarily false correlations, but you might get now negative correlations where there were none. So if you see now this distribution is shifted left, right? So now some of these are falling into the negative area where all these are positive. So that's sort of the implication there.
And it's also changing like your peaks.
Mhm.
Like you can see on that function how, yeah, the peaks themselves are coming down.
Yeah. So global signal has a bunch of influences on pretty much everything, as it should, because it's the global signal. So, okay, so what do we do about this? Glasser introduced this temporal ICA step or technique. So what they're going to look at now is temporal networks. In spatial ICA, what happens is we are trying to find spatial independence between voxels, and we're going to allow for some temporal overlap of the time series. In temporal ICA, we're going to look for temporal independence of areas and allow for some spatial overlap. So it's just flipping it. Conceptually, it's a little different. I'm still trying to wrap my head exactly what that means. But the idea here is that in ICA, or not in ICA, but in the brain networks are going to also have sort of specialized subnetworks. So if you have a major network, there could be just a part of that network that's involved in one process, part of that network involved in the other. And if they overlap spatially, spatial ICA is going to have a hard time picking that up. But temporal ICA is going to allow us to separate these temporal signals and have some sort of spatial overlap. Is that correct, Taylor? Am I saying that right?
Yeah. I mean, it's just, if you're using what they call ICA, you can use it. If you imagine, you have your data set up as your a matrix, right? You have on your columns the voxels, which are your variables, and then you have on the rows the time points, right? So the rows are the time points of your signal. Spatial ICA would be trying to make components as independent as possible among the columns. So imagine you estimated 100 components of voxels. They have to be as independent as possible. It's going to really split up all the spatial components into very fine-grained units. If you do that if you apply ICA on the rows, you get temporal ICA, which is trying to distinguish these. It's not doing independence in the spatial domain, like the voxels. It's trying to do it on the time courses themselves. So what happens is, the difference is, are you applying sparsity or independence in the spatial domain or in the temporal domain? If you apply it in the spatial domain, you get very sparse spatial components. If you apply it in the temporal domain, you get sparse temporal components, but the spatial ones are very, very global looking because you're not applying this part. I just went down a rabbit hole, but hopefully that makes more sense.
Yeah, so now we're gonna employ these two techniques to denoise. So spatial ICA is supposed to be really good at getting these structured sort of head noise components. So head motion in a structured way, regressing out those time series. Temporal ICA is going to be sort of good for these global fluctuations, I guess more related to respiration, things like that. So Glasser, this is one of the first temporal ICA papers, and here we're seeing temporal, they call them temporal modes or something, I'm not sure. But some of them look similar to ICA, some of them don't. The biggest difference here is that the positive and negative matters. One is not better than the other, whereas in spatial ICA, we only look at the positive usually. Temporal ICA both matters. And what you see are these interesting sort of I don't want to say anti-correlations, but brain areas that are sort of in opposition to each other throughout here. So you can see like a default mode here and then opposition. So these parts are negative, et cetera, et cetera. And then these are supposed to be two global signal components. I was trying to look for these in my data, couldn't really find them. But I think I need to do some more sort of quantification of the time series and things like that to identify them. But basically, they identified these two sort of global courses. And if you can see here, the two stronger lines are kind of going up. So a little bit more high frequency noise globally. Anyway. So this is showing their spatial ICA plus group ICA. So basically, they're doing ICA fix first on the individual subject level. And then they do a group ICA. They get those time series, so 137 time series. Then they do a temporal ICA on the group ICA, and then they get temporal ICA time series. And then you quantify noise and non-noise components, just like you would spatial ICA, and then you're going to regress that out of the data. I did a slightly modified version because there's a couple of different ways to do this, and I'll talk about that in just a bit. But these are the carpet plots again of all the voxels. So this is spatial ICA. So this is ICA fix here. And you can see we still have a lot of lines. Spatial ICA plus temporal ICA. So this is a temporal ICA cleanup. And then this one is global signal regression. So you can see that the temporal ICA cleanup gives you carpet plots similar to the global signal regression here. Same thing for all these subjects. So look at this, is very similar to this. They both clean the data set just as well from this. Same thing here. So temporal ICA looks similar to global signal regression. Both clean from that. And same thing on this subject here. So temporal ICA and global signal regression makes these carpet plots look similar. So they're advocating for the use of this spatial ICA plus temporal ICA denoising to sort of clean the data as well as global signal regression without regressing out any of the neural signal that we're interested in. And then they show us some of the benefits of this. What is this? I think this is a task or something. So you can see the difference that the temporal ICA is removing some of this global noise here. So it's just a little more negative in the contrast. And then temporal ICA is also pulling out this visual area in this task here. Whereas global signal regression doesn't really pull it out as well. So see the differences there. So temporal ICA denoising, this new technique to try and account for global signal noise while leaving the global signal intact. Okay, I'm not going to talk too much about this. We're just going to skip this, but basically looking at global signal topography, it depends on scanner site. So we see different scanners have different global signal topography. Here, we can imagine that's related to some kind of scanner noise or something. People have looked at differences in global signal topography and things like schizophrenia, depression, epilepsy, and basically you can see where the topography differs between control and clinical subjects. Here's an interesting one. So in the HCP data, they look at global signal topography according to rest or according to the task. And you can see that the task clearly changes the global signal topography here. So showing the influence of neural signal, global signal topography. Here's one where they correlate different things with different types of nuisance regressors. I'm not going to go too much into that. Okay, so getting to this study, how does global signal topography change across the lifespan? How do these different denoising techniques influence this topography? And how does global signal regression and these different denoising techniques influence lifespan functional connectivity here? So we had 601 subjects from the NKI resting state, 5 through 85 or 86, something like that. 1.4 TR, 10-minute rest scan, 299 volumes. And we did start with a low motion sample. So we cut out anybody with 0.05 average trim-wise displacement. I'm sorry, it should be 0.5. Well, yeah, 0.05 is ridiculous. It should be 0.5. That's like they'll be deceased, I think. They didn't really that much. So we still had age effects here. So age was linear, had a linear association with head motion. So the older you are, the more you move, sort of. Well, there's almost a curvilinear effect. I don't think we have so much power because you can see the young kids move a lot too, and then the 20-year-olds try to move the less, and then as we get older, so it balloons up there. So head motion was included as a covariate in our regression models. And this study is 60% female, so sex was also included as a covariate. So we looked at these different methods here. The basic pipeline was despiking, realignment, normalize, smooth, and then the modified pipelines. So one pipeline was just did ICA fix by itself, and then covariate regression filtering. One, we just did scrubbing by itself. One just did temporal ICA denoising by itself. And then one also did global signal regression by itself. So these are the main things that are going to change across the pipelines, whether it was ICA fix or just covariates by themselves, or was it fix plus the covariates, or was it temporal ICA plus all these covariates, or was it scrubbing in addition to these things? So there was a whole bunch of different combinations here. If scrubbing, so on average, each subject lost about 12% of their scan, which is acceptable according to power. We have two regression models looking at linear age and quadratic age. So first, we did ICA fix on the individual subjects, and then this is a group spatial ICA. across all 601 subjects. So this is going to be the first step of your temporal ICA, is you do a group ICA. Did it 125 components. Out of them, there were 39 noise. So even with ICA fix, what happened was we got rid of pretty much all the head motion noise components because there weren't any when I did the group spatial ICA. But there are still all these white matter components, which are related to physiological processes. So there were 39 of these noise components, and I regressed that out of the non-noise components. So we had 86 noise components that had all these regressed out, and then these 86 non-noise regressed time series went into the temporal ICA. And the temporal ICA, we had 75 components, and of those 9 were noise, 64 are deemed non-noise. I need to go back. I'm not really sure how to identify some of these yet, but you can see the networks here include things like the task positive. Here we have the salience network. And then we have really interesting ones where we have this thalamus positive related to this thalamus negative right next to each other. So there are a bunch of these in here. This one was tricky. I didn't know what to do with it. So it was brainstem negative and thalamus positive. So I'm assuming this has something to do with respiration because the thalamus is involved. So I didn't want to regress that out, even though it has this noise component in there. These are the ones I did end up regressing out. So they're brainstem stuff here. Mostly brainstem stuff. This one, like one of the few head motion components I kind of found. So there's some striping. Then we have like this brainstem noise. So most of them are like brainstem noise and stuff, which I think is good because I suppose it's capturing all these respiration or physiological artifacts. And then some I didn't know what to do, so I just kept them in, but I'm gonna take a look at these later. So looking at the relationship between these components and a global signal, this is essentially all the temporal ICA components, so 75 of them, and correlating the global signal from each subject with each component here. And then you can see some components are more strongly associated with the global signal than others. So for example, this one right here. So this component here is a motor component. So positive motor on one side, negative motor on the other side. You can see that's pretty strongly related to global signal. Here's another one here, which is our task positive. So this area here. This one here is also another motor component. And the noise components are actually not that strongly related to the global signal. So if you see this one, the second column here, it's not so dark. Is that right? Oh no, I can't see red, so I'm hoping this is correct. So this one right here is a noise component. In this column, you see there's not so much color there. So it's not so strongly related. This one is a noise component there. So a lot of the non-noise components seem to have stronger relationships to the global signal than these noise components here, if I am reading my colors correctly. So that's good news, showing that the temporal ICA is sort of decomposing these time series, and also that the global signal is made in large part of non-noise components. So these neural signals are contributing a lot to the global signal. Seems like more so than these non-neural signals. So that's good for my story. So let's look at what happens to the global signal as we clean the data. So we have 3 subjects here. This subject has very low motion, 0.08 on average. You can see he hardly ever moves at all. This guy's like a rock star. This guy, 0.28 on average. So this person has high intermittent head movements. And this person, 0.49. This person just has high movements the whole time. So we have 3 different types of head movement subjects here. Low head movement, sort of intermittent large motions, and then here just sort of constant large motions here. This is the frame-wise displacement here on the top. So correlating the global signal with just covariate regression with our spatial ICA, so the ICA fix. And if the subject is low motion, you see there's a pretty high correlation. So ICA fix doesn't really affect the global signal that much. But if they have these larger types of motions, then ICA fix, the correlation goes down to 0.9, still pretty strong. But obviously having a much greater influence if the subject has a lot of motion. And then here we have the temporal ICA time course in yellow. And if we compare that with just the covariate, we get 0.78, we get 0.80 here, spatial ICA with the temporal ICA. So the temporal ICA you see is taking out a lot more of the signal here. And that increases if the subject has bad motion. So you can see the subject with the worst motion here. The correlation between the global signal is the lowest sort of here. So showing that the temporal ICA has a much greater influence on these high motion subjects. Interestingly, there's a huge range here. So I need to go and look at these subjects that only have a 0.2 correlation. So obviously temporal ICA is doing something really severe to these subjects here. Okay, so showing that temporal ICA is influencing the global signal. So some carpet plots, again, the same subjects, low motion subject, large intermittent movements and large constant movements. Something is wrong with the scale here. I need to go and influence that. And these are also flipped the wrong way. So, excuse them, supposed to be flipped the other way. But it doesn't matter. You can tell that the temporal ICA is doing just as well as the global signal regression here, which they both clean up from this one. So it's covariates only. This covariate plus global signal, ICA fix plus the global signal, and then temporal ICA. So you can see temporal ICA does as well as the global signal regression. Yeah, so showing the same thing that Glasser did. So hey, good news, temporal ICA is doing what I hoped it would for this data set. Let's look at global signal topography across the lifespan. So this is correlating the global signal with each voxel in the brain, and then correlating those functional connectivity strengths with age. Looking at linear trajectories first, so we can see the global signal. Oh, there's some masking problems here, so pay no attention to these missing parts here. But essentially, so the same thing, no matter what type of denoising, it seems like the global signal, spatial topography across age, is related to neural signal instead of noise signal. So no matter how we clean it, we still get this linear increase in the thalamus and these attention areas here. For the linear one, you can see it's doing a good job of taking out the noise. So I always kind of thought this was noise because it looks like noise, sort of here in the mid sagittal slice between the two hemispheres. So you can see that covariates don't get rid of it. Fix and scrubbing doesn't get rid of it. Fix doesn't get rid of it. It's only the temporal ICA that gets rid of that noise over there. So that's good. Quadratic effects. So the lateral frontal parietal network, or the central executive network, the frontal eye fields from the dorsal attention network, you can see that those stay intact even with temporal ICA cleaning. So that's good news. This one doesn't really change that much. So frontal and visual areas stay intact the whole way through. So that's good news. So this is showing sort of that the global signal topography has a distinct trajectory across the lifespan. So certain brain areas are more strongly associated with the global signal at certain points in life. And no matter what type of cleaning, that pattern still stays, suggesting that that is neural activity and not noise contributing to this pattern here. So I submitted this first without any of this cleaning, just a fix in the scribbling. And the reviewer didn't like that because they said I didn't know if it was neural or non-neural. So I did these other ones, and hopefully that's showing that this is related to neural activity from the global signal here. So that was the point of this analysis here. And tell me if you have any questions. I know there's a lot of data here. So looking at ROI to ROI, I just did one analysis, global signal versus no global signal. And you can see that global signal has a pretty heavy influence here. So linear connections. If there's a color, that means it's significant, FDR corrected, that little box. If it's white, that means there's no effect. And you can see that there are much more significant effects after global signal regression compared to before it. And then you can see it also changes the nature of some of these correlations here. So going from a negative relationship here to a positive relationship here. So I think that's sort of going along with that shifting correlation histogram we saw. So it's shifting these correlations. And then here's pushing them more negative. You can see things like that. Okay, so the next thing I wanted to do was look at how does global signal regression influence ROI to whole brain connectivity? So this is ROI in the thalamus. We get the thalamus seed, we correlate that with every other voxel in the brain, and then we do this association with age again, so across the lifespan. So the thalamus here, if we just do covariates, there's just a little thing going on here. Scrubbing doesn't help it so much. Covariates, scrubbing plus global signal, and then all of a sudden the default mode pops up. So something really drastic is happening here with global signal regression and the thalamus. It doesn't happen for fix, but it does happen for fix plus global signal regression again. So global signal regression is definitely doing something, and nothing comes out for temporal ICA. So this makes me think this is one of those things where it's pushing the correlation matrix to one side, because it's not showing up in the temporal ICA. This one, negative relationships, I'm thinking this one is noise. You can see here that global signal regression gets rid of some of it. So it makes it this nice little anterior cingulate here instead of this whole big area. And you see that temporal ICA doesn't get rid of it, but I think that's because I didn't regress it out. So there is a component that looks exactly like that I didn't regress out. So if you look at this guy right here. So I kept that in, but to me it looks like neuroactivity because it has this sort of cingulate and then the frontal eye fields juxtaposed with the parietal areas. So I don't know if that's noise. I need to do a little more investigations there. But that's that. And that's why I think the temporal ICA didn't take care of it, because I left that component in there. Okay, positive quadratic. We see that covariates for scrubbing doesn't really do anything, so scrubbing doesn't add a lot. Global signal regression changes this now from the frontal eye fields to the lateral sensory motor areas, which is interesting. And then you can see the frontal pattern changes also. Let me get this temporal thing now. Fix plus global signal regression. So sort of the same pattern here. And then temporal ICA gets us this. These guys right here. So again, is this from shifting correlations? I'm not sure, but the temporal ICA is giving us a much different pattern than the global signal regression there. Sort of same thing here for the negative quadratic. Global signal regression again is pulling out this and this here, sort of by the thalamus in here. Temporal ICA, nothing. So is that possibly noise? Temporal ICA gets rid of that, then it adds this little visual area here. Okay, One more, so now looking at the central executive network, so I took a node in the in the frontal area here. Oh, no, in the parietal area here, so around here somewhere in the central executive network. And then looking at these again, you can see that temporal ICA is going to give you something a little bit smaller than the global signal regression again. And global signal regression is similar to scrubbing and fix. So not much influence of global signal in this, but the temporal ICA sort of gets rid of that. Temporal ICA sort of brings out this activity too here in the red compared to some of these other ones that didn't come out with global signal regression. Then for the negative one here, same thing sort of happens. So scrubbing gets us this, which doesn't happen, just chloride. So here scrubbing is helping. Global signal gets rid of it. ICA fix plus scrubbing brings it back. Global signal plus fix plus scrubbing puts it back like this. And then temporal ICA gets us this. These networks here. And this effect is much stronger than all of these here. So makes me a little more confident this is actually a neural signal. And then same thing for the negative quadratics here. So we see the temporal ICA is giving us a good clean here. Similar, well, not so the global signal is doing something different than temporal ICA clean. And then same thing here, where you can see global signal regression still has all this noise here in the brainstem, thalamus area here, but temporal ICA gets rid of all of that. Okay, so we see a distinct GS topography across the lifespan, despite what type of denoising technique I use, suggesting that the global signal or the neural component of the global signal is what is driving these associations. These different techniques, oh, that's what I just said, little influence on GFC lifespan topography. And then we also see the influence of global signal regression and temporal ICA. So temporal ICA seems to be doing something much different than global signal regression, even though the carpet plots come out the same. So something is obviously happening where temporal ICA is giving us something that global signal regression doesn't. And hopefully that is temporal ICA is allowing us to keep the neural component of the signal, global signal, while regression out everything else. And that's it. So, any questions? Yes.
Yeah, I think it's like before you went into the current study, when you're talking about the temporal ICA, and then you mentioned like the one study had like it looked like what temporal ICA you got, like a global signal.
Wait, which one?
I think it was
I'm sorry, I can't hear anything.
oh yeah. I think it's a blaster. But you're going through temporal ICA and then basically, yeah, that one, and then you get like the global signal
This one?
on the other side.
Oh, this one.
Yeah,
Here?
yeah, I'm just a little like, yeah, so you get, so like you can potentially get that with temporal ICA. So I'm just like trying to wrap my head around like what that would mean to like if you would you keep that in if you were to get it in your current study. I know you said you didn't.
I didn't find it. I have to go look at what Glasser did. I think they say that this is noise here. This is just a global fluctuation unrelated to neural activity, and I think they regressed that out. You have to look at this. This is the one where they
Okay,
actually
I'll
did
look
it.
at it. Yeah, it's just something like even with our like when we do the normal IC, sometimes we get like that global signal component. And it's like what do we do with it?
Yeah.
And then, but it does seem like there is a difference. Obviously, with global signal, it's like the whole brain, where it's like you might get a few components like that, and it's not as detrimental, maybe.
Yeah. So I was actually interested in seeing how that's gonna work out.
Okay.
So
Okay.
if we can apply a cap analysis to this, and
Yeah.
maybe we don't get those global components, then that temporal ICA is gonna be pretty handy for us.
Okay.
So yeah, that's definitely something I'm interested in.
Okay, cool.
Let's see.
Yeah, that was like just my question with that.
All right, Taylor, thanks, man. Anything else? This is kind of a
More meta.
heavy talk. Yes,
How do you keep track of all these analyses in your head?
spreadsheets. Well, not spreadsheets. I put it down in the slide and then I do the analysis and fill it in. So,
Okay, yeah, that makes sense.
but yeah, I got kicked off the server a couple of times. So it's using
Yeah.
an insane amount of processing power. But anyway, anything else?
Oh, and then, sorry, just for the template ICA, I'm guessing you use like a toolbox.
No, I used a Python package from Taylor.
Oh, cool.
I want to look at the GIF toolbox though, because I think they have it, it's called Fast ICA, they call it.
Okay.
So that might be one. There's also a MATLAB package that's what Glasser used. So instead of Glasser's paper, they point to a MATLAB package. But Taylor has a code for a Python temporal ICA that we can use in the temporal ICA.
Wow.
So it takes, you have to do the spatial ICA first, then
Okay.
get the components, regress out the noise components, then feed those into the temporal ICA. And then you find noise components again. And then you regress everything out of the data after. The spatial ICA noise components, the temporal ICA noise components, the first and 24 regressors all at once in the matrix. So I have scripts for all this, so we can apply it.
Awesome.
So, yeah. Anything else, anyone? All right, I know it was a pretty dense talk, so you guys can think about it.